All living things are made of tiny structures called cells. A cell is the smallest unit that can perform all the processes necessary for life. All cells are surrounded by a cell membrane. Structures floating in the fluid inside this membrane are called organelles. They perform specific functions within the cell. The most important organelle is called the nucleus. It contains chromosomes.
passed on from our parents in these chromosomes, which are really just condensed, coiled up molecules of DNA. DNA has a specific structure called a double helix. The double helix describes the structure of a ribbon on one side with extensions coming off of it internally, attached to a mirror image of that ribbon on the other side, bonded to the extensions coming off internally. And that creates a twisted structure called a double helix. The double helix of DNA consists of a molecule called deoxyribose, which is a sugar, a phosphate group, and then these extensions called nucleotide bases. And the specific nucleotide bases are called adenine, guanine, thymine, and cytosine. Those are the nucleotide bases you find in DNA. When we move to this picture, we can see more specifically the double helix with the nucleotide bases labeled. The interesting thing and unique thing about these nucleotide bases is that they will bond to other nucleotide bases using a hydrogen bond. But the important thing that makes DNA so useful and makes our protein synthesizing somewhat predictable is that these nucleotide bases will only bond to another specific nucleotide base. For example, adenine will only bond to thymine in DNA. Guanine will only bond to cytosine in DNA. If you look at the molecular structure in the middle, you'll see that we have the backbone, which is sugar, the deoxyribose, and phosphate, and then attached to that, going towards the other side of the double helix, is the nucleotide base, cytosine, here.
There are 4 true stages of the cell cycle: G1, S, G2, and M. A 5th stage, G0, is where the cell remains indefinitely until it begins the cell cycle again. Some cells may never leave the G0 stage. Others that go through division rapidly, such as bone marrow cells, may skip the G0 stage entirely. Cells begin the cell cycle by entering interphase. In interphase, the cell prepares for division by duplicating DNA. Interphase includes the stages G1, S, and G2. In G1, the cell synthesizes DNA and protein and increases organelle and cytoskeletal elements. The chromatin consists of long, slender rods jumbled together in the nucleus. S is the synthesis stage. Nuclear DNA replicates along its centrioles, which are small structures made up mostly of microtubules that associate with the chromosome's DNA. The chromatins begin to coil so that they shorten and thicken. In G2, RNA and protein are synthesized. Proofreading also occurs. The DNA replicated in the S phase is checked for mistakes. If mistakes are found, the cell repairs them or receives a signal to self-destruct, called apoptosis. The difference between G2 and G1 is that in G1, DNA and protein are made, while in G2, RNA and protein are made. In the M stage, the cell divides into 2 identical cells through divisions of the nucleus, mitosis, and cytoplasm, cytokinesis. Over 5 different stages, the centrioles pull the chromosomes apart and the cell divides in 2. First, in prophase, the duplicated chromosomes appear and then split into identical halves called chromatids, attached by spindles called centromeres. The nuclear membrane disappears and spindle fibers form in the cytoplasm, radiating from the two centrioles located at opposite poles of the cell. In metaphase, the spindle fibers pull the centromeres, aligning them in the middle of the spindle or the equatorial plate of the cell. The two chromatids are clearly visible in this stage. In anaphase, the centromeres split, pulling sister chromatids to opposite sides of the cell. By the end of anaphase, 46 chromosomes lie at each side of the cell. When chromosomes are separated, each sister chromatid is considered a new chromosome. In telophase, a nuclear membrane forms around a group of 46 chromosomes. The spindle fibers disappear and the chromosomes uncoil. Cytokinesis occurs and the cytoplasm divides into equal parts. Two identical daughter cells have been formed. Each new cell moves back to the G0 stage and is ready to begin the process again.